Please. Thank you very much for coming again, please. Now, the last areas please to be discussed: distal tubule and collecting ducts. We have completed the journey of the proximal tubule, loop of Henle. We know what happens to the fluid after it passes through loop of Henle. You have absorbed the solutes, but you are not reabsorbing the water, and that's why the fluid has become diluted and the osmolality is decreasing, and it's a hypotonic fluid which is entering into the distal tubules. Okay? Now we are beginning. What happens next? Distal tubule. Okay? Well, distal tubule is divided into two parts. The first part is early distal tubule, and the second part is the late distal tubule. Okay? In the early distal tubule, you have a very special thing. You have done it. It's macula densa. Do you remember that? For the auto-regulation of GFR, it is modular in the beginning of the distal tubule. Yani, it is present in the early part of the distal tubule, first half. Okay? Well, early distal tubule is also behaving like thick loop of Henle in the sense that it is not reabsorbing water. Okay? It is not permeable, almost impermeable to water. Okay? And also it is not reabsorbing the urea. Urea and water are not reabsorbed from these areas. There is some reabsorption of sodium chloride and some other ions normally, no problem. But what you are not reabsorbing is water. Okay? If you are not reabsorbing the water and you are still reabsorbing the ions, what will happen to the hypotonic fluid which is coming? It will become more hypotonic, right? Because you are removing water, reabsorbing water, not removing water, and you are removing ions only. So if the water is getting reabsorbed, then situation would have been different. But this segment is just like taqriban like the thick loop of Henle. No reabsorption of water. When there is no reabsorption of water, water is staying in the lumen, ions are going out, the solution is becoming more dilute, so it's hypotonic solution. Okay? Hydrogen ions are secreted still from this part also. Hydrogen ions were secreted from the proximal tubules, they were secreted from the thick loop of Henle, and they are now secreted here by secondary active transport. Okay? The same sodium hydrogen counter transport. Remember? Hydrogen ions. Okay. In the early distal tubule, there is a very special transporter. Do you see that? Okay? This transporter is called the sodium chloride co-transporter. Okay? Now look at that please and answer. What do you think about this one? It's a tricky one. Sodium chloride co-transporter attaching to the carrier. Two ions are moving inside. Both are going from ECF to ICF. Think about them and answer, where should I place it? Shall I call it active transport or the passive transport or what? Okay, are you in favor of passive? If you are saying passive, why are you saying passive? Okay, concentration gradient. Both are using their concentration gradients from high to low. Chloride and sodium, from high to low they are moving. Okay, but this is going by attachment. So what does it become? It is known as the facilitated diffusion, isn't it? So both are showing the example of facilitated diffusion. On the other side, on the back side, a normal pump is present. What is that pump? It is present everywhere. It is present everywhere, nothing special. This sodium chloride co-transporter is clinically and pharmacologically important. You can block it by a special group of diuretics, not the loop diuretics because it is the distal tubule. These are thiazide diuretics, another very, very famous group of diuretics. Do you remember that? Okay. So thiazide diuretics block this transporter and more sodium chloride and water will stay in the lumen and will pass in urine. Okay. This is about early distal tubule. Late distal tubule, please. Now the region is changing, the characteristics are changing. Early distal tubule is more or less like the thick loop of Henle because it is not reabsorbing the water. Okay? So this is called the diluting segment. It is a diluting area because it dilutes the fluid. Early distal tubule, diluting segment. Thick loop of Henle, also diluting segment because it is not reabsorbing the water. Okay? So these two areas are typically diluting the fluid. Which two areas? Thick loop of Henle and early distal tubule. Alright? Okay. Look at the late distal tubules. Two special types of cells are present. One regular cells called the principal cells. I don't know if you have done it in histology or not. The principal cells and intercalated cells, have you done it? Histology. You're not finished with histology? Are you done? Okay. Alright. Principal cells and the intercalated cells. Principal cells are the regular cells, which are the cuboidal cells lining the tubules, normal cells. In between them, here, there, about 10% cells are present. These are dark type of cells, okay, containing granules and special cells. They are called the intercalated cells or the dark cells. So the P cells are the normal cells and the dark cells are the intercalated cells. So you will find two types of cells in late distal tubule. This type of pattern will continue in the collecting ducts also. Yani the structure of collecting duct and the structure of the late distal tubule is kind of similar in form of cells. Okay? Two types of cells are present, regular cells, P cells, and some special cells, intercalated cells, the dark cells. Okay? Alright. Late distal tubule reabsorbs the salt and water, definitely, but even then, if both salt and water is reabsorbed, okay, the fluid still remains a little hypotonic. Okay, not as hypotonic as before, but still, because salt and water are moving, so it will remain hypotonic. Okay, so what is the status of the fluid in the late distal tubule? It is still hypotonic. Okay, it was hypotonic in the upper part of thick loop of Henle. It was hypotonic, more hypotonic in the early distal tubule. Now it is in the late distal tubule, but it will still remain hypotonic. It is not becoming isotonic yet. Okay, it will become isotonic soon after, inshallah, when it will go into the collecting ducts. All right. So we are going station by station. We are moving and seeing what is happening to the osmolarity of the fluid. All right. Okay. Principal cells, please. Principal cells are the regular cells found in the distal tubule, late distal tubule, and the collecting ducts. And in the late distal tubule, one hormone is acting. Well, from late distal tubule, remember one thing. Now it's the area of the hormones. Your two very important hormones will begin their action from late distal tubule. Till early distal tubule, you are not talking about these hormones, but now you are naming two special hormones which are very important for this area. One is aldosterone and the second one is ADH. Two are very famous. Get me very quickly the functions of the aldosterone. If aldosterone is working in this region, what will aldosterone do? What will it help? Sodium reabsorption. Very nice. Along with sodium, what else? Water. Okay. There is one friend of the sodium which is always try to chloride. Sodium reabsorption, chloride reabsorption, and together with these two, water will follow. So three substances or three things are getting reabsorbed. Sodium, chloride, and water. Sodium is reabsorbed, chloride is going along, and water will be going along with these two. Okay? Two things are getting secreted in this area. Secretion, opposite direction. Secretion now. Potassium is secreted, please. Potassium is secreted, one. And one more ion, which is hydrogen, everywhere, majood. So now you have a set of 5 functions of aldosterone. Can you quickly recap and call 5 functions of aldosterone? 3 reabsorption, 2 secretion. Number 1, sodium, chloride. Great. Now about the secretion. It's 5 now. Thank you very much. Great. Okay. This is done by aldosterone. ADH has only one function mainly. okay, which is the reabsorption of water. When the ADH is there, ADH will make special channels, okay, in the wall, in the cell membrane. Okay, if ADH is there, for example, if this is the cell and this is the lumen, and if ADH is working here, ADH will make special protein channels here, the new channels, and these channels will allow the water to pass through. If the channels are not موجود, water will not be reabsorbed from this area. If the ADH is there, it will form the water channels. These water channels have a special name, please, okay? Aquaporins. What do you call them? Aquaporins. Aqua means water, porins means channels. Aquaporins are the water channels which come, which appear, which are attached when ADH is there, okay? Without ADH, khalas, they are gone, ghaib, they are no more. When ADH is there, they will appear. When ADH is not there, they will disappear, okay? So these are aquaporins, the channels. All right. Principal cells are the regular cells which are reabsorbing salt and water. Aldosterone is acting on that, ADH is acting on that, okay. Intercalated cells, special cells, less in number, dark in color, condensed cells, contain some granules, and their function is special also. What do? The intercalated cells have a very special function. They are secreting hydrogen ions, they are producing hydrogen ions, but they are producing hydrogen ions in a very different way, okay? So far you did the hydrogen ions from everywhere, secretion, proximal tubule, thick loop of Henle, early distal tubule, late distal tubule, everywhere there was, okay, you will see secretion, regular secretion, sodium hydrogen counter transport. But now you are focusing new type of cells. These cells are intercalated cells. They will not show you the secondary active transport. They will show you something different. Do you see a transport of hydrogen ions here from the lumen? Do you see the picture? Can you see? What kind of transport is shown here? Primary active. And why do you call it primary active? ATP is there, okay? So if it's primary active, shall I call it hydrogen transport or hydrogen carrier or hydrogen pump? So this is a hydrogen pump, primary hydrogen pump, ATP directly used from high concentration to low concentration or from low concentration to high concentration. What is your idea? It is primary hydrogen pump, okay, from low to high, okay, it is pumping against low to high. These are the special cells, okay? So remember, intercalated cells are secreting hydrogen, but these hydrogen ions are coming by a special mechanism. Primary hydrogen pump is there. That primary hydrogen pump was not present in the proximal tubule. That was not there in the loop of Henle. That we did not see in the early distal tubule. Here, yes, it is there, okay? So hydrogen ions are secreted in the nephron by two mechanisms. What are these two likely mechanisms? Number one is secondary, and number two is by quantity. Which mechanism would be secreting more, logically speaking? Primary or secondary? Secondary or primary? Primary? Quantity wise more. Logical, very logical. Secondary or primary? Secondary, because look at the number of cells. Proximal tubule, thick loop of Henle, early distal tubule, late distal tubule, and also collecting ducts. And all areas are secreting secondary hydrogen ions by transport mechanism, okay? Primary is khususan coming from only few cells, only 10% of the cells. So it's a very powerful process, but again, the number of the cells for secondary active transport is more, okay? So by quantity, the bigger quantity of hydrogen is secreted by secondary and less quantity is secreted by primary, okay? All right, okay. At the same time, if you see what is happening inside the cell when the hydrogen is secreted, at the same time something else is happening. Look at the chemical reaction happening in the cell. Look at the chemical reaction. Inside the cell, carbon dioxide and water are combining, okay? And there is a very special enzyme called carbonic anhydrase. Wherever this enzyme is موجود, it will do special thing. It will combine carbon dioxide and water, it will produce carbonic acid, and it will cause HNHCO3 like this. So do you see? Hydrogen ions are formed, and at the same time bicarbonates are also formed. Hydrogen is going out of the cell, secretion, but bicarbonate is going by the back door. It is going out of the cell, base of the cell. But bicarbonate is using a very special transport. What is it using? Antiport. Okay, it's an antiport type of transport, opposite. It's counter transport. One chloride and one bicarbonate is exchanged. Okay, so one chloride is going and one bicarbonate is coming out. Going inside the cell is chloride and coming out of the cell is bicarbonates. Okay, so this is a bicarbonate chloride transporter. Will it be a secondary or primary active transport? Very simple. Secondary. Okay, you will say secondary. Why will you say secondary? Because it's counter and, well, if you say counter, sodium potassium pump is also counter. You can't say on the base of counter. Say something else. Okay. Why? Yeah, because you don't see ATP maktoob here. Okay, you don't see any direct ATP, do you? No, you don't see ATP. It means it's going in this way. Okay. Chloride and bicarbonate. Bicarbonate is going out and chloride is coming in. All right. Which one is going against the concentration gradient? Bicarbonate or the chloride? Very simple. Bicarbonate. So active transport of the bicarbonate ions. Okay. Good. That's a nice choice. And all right. Collecting ducts. Now you are coming down in the collecting duct, same type of cells, principal cells and the intercalated cells. You are having absorption of salt and water. Aldosterone and ADH on the collecting ducts, they are working. Okay, aldosterone is working and ADH is also working in this area. Especially, collecting duct has two parts, the upper part and the lower part. Okay, the upper part is the cortical part and the lower part is the medullary part of the collecting duct. Okay. Aldosterone, ADH, both are working on the cortical part. All right? Cortical collecting duct, please. What are you doing? You are now reabsorbing water more than the solutes. Now you are reabsorbing water more because ADH is working very well here. If ADH is working, more water is reabsorbed. If more water is reabsorbed, Shabab, what will happen? Previously it was hypotonic. Now after coming in this area, what change will come? First isotonic, definitely. So the fluid will become isotonic because you are reabsorbing the water more. Okay? So water because of ADH. Alright. Some secretion is there. Secretion is expected. You will easily say secretion of hydrogen. You will say secretion of potassium ions. And in the beginning I told you one more thing is secreted. In many areas, ammonia, remember, I told you ammonia in the beginning. Ammonia is also secreted. Shabab, remember one point. Keep it in mind. It may not be written in the book, the new edition, but it's a fact. From proximal tubule to the last part, collecting ducts, okay? Hydrogen ions and ammonia, they are produced and secreted by all the areas except two. All the areas except two. And these two are the thin descending and thin ascending loop of Henle. Just leave the thin portion. Just leave the thin portion. They are not secreting urea, they are not secreting ammonia, and they are not secreting hydrogen ions, okay? All the cells are giving hydrogen and ammonia in different quantities, okay? So if I say, what is the exception? You will say thin portion of the loop of Henle bus, thin descending and thin ascending. That's it. So every area is giving you all these things. Fluid is isotonic. Now you are in the last part of the medullary collecting duct. Okay? ADH is very powerfully working here. More receptors of ADH, more function. Aldosterone function is now not seen on the medullary. So now very quickly you call. Aldosterone is working on the late distal tubule, yes or no? Aldosterone is working on the cortical collecting duct, yes or no? But aldosterone is probably not working on the medullary. Okay, so two areas where aldosterone is working. ADH is working on the late distal tubule, yes or no? ADH? Yes, Definitely. ADH is working on the cortical collecting duct. Yes. ADH is working on the medullary collecting duct. So which area is the bigger area? ADH area or the aldosterone area? ADH. ADH is working on 3 parts actually, and aldosterone is working on 2 parts. Okay. So because of the receptors which are present here, so in the medullary area, large amount of water is reabsorbed because of the ADH action. If you are reabsorbing the water, what will happen? Now the fluid from isotonic side will turn into hypertonic. If you have more ADH, the urine will become more hypertonic. If you have less ADH available, it will become less hypertonic. It depends on how much ADH is modued in the blood. It depends on ADH. So ADH is the reason why water is reabsorbed from the collecting ducts, okay, and from the late distal tubules also. ADH is the main water reabsorbing hormone. One more thing. In the medullary collecting ducts, when ADH is working very well, it's great. So large amount of water is reabsorbed and the fluid becomes hypertonic. The concentration of substances increase because the water is gone. Okay? The concentration of urea will also increase, right? Because everything is increasing, so urea will also increase. When the urea concentration increases, urea now starts moving out of the medullary collecting duct and it comes into interstitium. Okay? So urea is reabsorbed from the medullary collecting duct only when ADH is present, because ADH will cause water reabsorption. After water reabsorption, the concentration of the urea becomes very high in the medullary region, and from the medullary region now it's permeable to urea and it will reabsorb the urea. So the primary action of the ADH is to reabsorb water. But what will be the secondary effect of that ADH? Urea reabsorption. It's not the primary one. Because of the water, secondarily you will have the urea reabsorption. So urea is reabsorbed significantly from this region. Okay? You might think, why would urea be absorbed? What is the use of urea? It's a waste product. Why are we doing that? The role of urea will come. It is circulating in the interstitium. It is again entering, coming out, again entering. So urea is circulating between interstitium and the tubules. Half of it is passing in urine and half of it is moving and circulating between the tubules and the interstitium. And we'll see why this urea is important, why it is circulating, and what is it doing. Is it really helping us, or doesn't it have any function, or does it have any significance? We'll answer this question in one of the lectures. But remember, urea is reabsorbed significantly from medullary, not from cortical, not from late distal tubule, from the medullary, because the concentration now really increases. Okay? So now you know the hormone dependent areas of the tubule. Late distal tubule, cortical collecting duct, medullary collecting duct. Now you are comparing 2 main hormones, ADH and aldosterone. ADH is working more, 3 areas. Aldosterone is most likely working on 2 areas, upper 2 areas, and ADH all 3 areas. Did you see that? Okay. All right. Okay. I am right now leaving this regulation of the tubular reabsorption, okay, and this term which I will explain to you later, glomerular tubular balance, because I want to save time, and one slide of reabsorption, some hormones with their action. Actually it is written, you can read it yourself, what are these hormones doing, aldosterone, angiotensin 2 on the tubules, what is their action, ADH, atrial natriuretic peptide. We did not mention this hormone, we'll read it inshallah, and some other hormones are acting on the tubules. The parathyroid hormone is acting for calcium, cortisol is acting, it's testosterone and estrogen, progesterone, they are also acting in minor amounts. Our major idea was to discuss the main hormones which are working like ADH and aldosterone. Okay? We also did the function of angiotensin 2 previously, if you remember. Okay? Alright. So we are holding this discussion right now till here.